Mr. Sylvester.
Thank you. Basically, I got started looking at tocotrienols in cancer when I worked at Roswell Park Cancer Institute, and we were studying fat in cancer, and one of the fats I was looking at was palm oil, and we just stumbled on the anti-cancer effects of palm oil against breast cancer. Now, cancer, as you know, is kind of an umbrella term. It represents more than 100 diseases, and that are characterized, they have similarities, but they're all separate diseases, and it's characterized by having uncontrolled cellular growth, tissue invasion, and metastasis. Now, the current strategies, the old-fashioned strategies, were to have something that was toxic and just killed cells rather non-specifically, and the new strategies are developed now to specifically target what the mutations or receptors or the enzymes that are dysregulated in a cancer cell. And you develop drugs that are directed against these specific molecular targets, and you get a very specific and a very effective way of inhibiting these tumors' growth and progression. Now, the beautiful thing about tocotrienols is they have great potential in the prevention and treatment of cancer because they have so many multiple mechanisms of action that are directed against tumor cell proliferation and viability. And we've already talked about many of those different mechanisms in terms of enzymes and receptors and that sort of thing. So it's a great opportunity to exploit this action of tocotrienols. Now, the way I got started in this, we were looking at the anti-proliferative effects of tocotrienols against breast cancer, and we found that after many experiments that tocotrienols inhibit growth factor receptor activation. And as you know, these growth factors are involved with turning on mitogenic signaling pathways, and if you shut those off, you block cell cycle progression and basically inhibit cancer cell division. Now that's at low doses. At high doses, the tocotrienols are actually cytotoxic and induce apoptosis. Now, I'm just going to review quickly what happens with these growth factor receptors. In our initial work you looked at the epidermal growth factor receptor, or EGF receptor. In human, it's human epidermal growth factor receptor, or HER. In the mouse, a rodent, it's ERB receptors. But basically, the way this represents two receptors, and basically EGF will bind to the receptor, and this will cause a dimerization. And then what happens is the cytoplasmic region of the receptor is an enzyme. It's a receptor tyrosine kinase, and as a result, it'll bind ATP, and one side of the receptor will phosphorylate the contralateral side of the receptor, and this will continue, and you have these autophosphorylation sites right here. Now, what's important about those autophosphorylation sites is that those are absolutely essential for the receptor to interact with its substrates. And so if you block this autophosphorylation, what happens is the receptor can't interact with its substrates, and you prevent signal transduction. And this is basically when you have the signal transduction, it does everything from growth, proliferation, survival of the cell and the tumor cell. Well, early studies we did looked at the HER receptor, and we looked at various compounds. Now, we knew tocotrienols, these are just growth studies, tocotrienols will induce a dose-response inhibition of mammary cancer cell growth. The other two drugs here are erlotinib and gefitinib. These are HER inhibitors that affect the ATP binding site and inhibit ATP binding, so it prevents the phosphorylation. And then trostimid is an antibody that basically blocks the activation of the receptor. Now, what we found is gefitinib and erlotinib and tocotrienols show a nice dose-response inhibition in preventing or inhibiting tumor cell growth. Now, this is in culture. And what we did, and if we go back here, We used very low doses of tocotrienol and uratinib. In this case, we used ineffective doses like 2 nanomole or micromolar, and then for gefidinib and uratinib, we used 0.25 micromolar doses. And this is really where we did a combination study, and we were looking at very low doses, very, very low doses. As you can see in when the treatments were given alone, this would be control. This is tocotrienol, uratinib, and gefidinib. These are these sub-effective doses. But when you put these 2 sub-effective doses together, you see this tremendous inhibition in cell growth. So you take very low doses of the 2 compounds, put them together, and you see this great inhibition of tumor cell growth. And this is just showing Ki-67 staining, where Ki-67 is a protein that's expressed in the nucleus of actively proliferating cells. And it shows up, and I'm not sure if you can see it very well, but it's these pink dots. That's the positive staining. And as you can see in the control and the single treatments of uratinib, gefidinib, and tocotrienol, you see that there's really not much difference from the untreated controls. But when you put the HER inhibitors together with tocotrienol, it totally eliminates the Ki-67 staining. In other words, you inhibit cell proliferation, you inhibit cell cycle progression and cell cycle division. So this showed that it was very potent. By using very low doses of these compounds, you can inhibit tumor growth. Now, what we had shown earlier was that if you look at the HER family of receptors, there's 4 in that family, otherwise known as HER1, HER2, HER3, and HER4. In the mouse, it's ERB1, ERB2, 3, 4. What we looked at was the same doses and effects. These are a metastatic, highly aggressive mouse tumor cell line. And we looked at the doses on the expression of HERB3 and HERB4. Now, one of the interesting things we found was that tocotrienols really don't affect HER1, and they have a little bit of effect on HER2, but their biggest effect is suppressing the activation of HER3 and HER4. And as you can see, when we were using the sub-effective doses, there's really not much difference in any of these groups. But when you put the 2 together, these very low doses, you see this inhibition of not only the level of HER3 or ERB3 and ERB4 receptor levels, but the autophosphorylation. And like I said before, the autophosphorylation is absolutely important for signal transduction. So here it is, you take these sub-effective doses, and you get a very nice inhibition of receptor activation and actually downregulate the receptors. And this is just some cytochemistry that we did for ERB3 and ERB4. And this is an antibody that was directed against the phosphorylated ERB3 and ERB4. And as you can see, and this is with a control, tocotrienol, and then the combination of uratinib and tocotrienol together. As you can see with ERB4 and 3, there's nice expression, it lights up, but when you put the 2 drugs together, you block that phosphorylation and expression in the cells. So our initial studies were like I said, most of them were in looking at the HER receptor, or EGF receptor, which is, like I said, a tyrosine kinase receptor. And there's another type of receptor that's a tyrosine kinase, and that's the MET receptor. And the MET receptor is very interesting because it's associated with changing cellular characteristics. Cells that have aberrant MET receptors sometimes this is a defect in many types of cancer, are very aggressive and metastatic. Now, MET is a receptor for hepatic growth factor, HGF, otherwise known as scatter factor. And the reason it's called that is because most epithelial cells are very localized, they stay put and don't move. But when you stimulate them with HGF, they start changing their shape and they start moving, and they can be quite invasive. So HGF is a growth factor normally active in wound healing, liver regeneration, organ morphogenesis. So it's one of these growth factors that changes the morphology and behavior of cells. Now, when it's dysregulated, it results in increased cellular proliferation, survival, decreased adhesion to the basement membrane. These are in epithelial cells. Cytoskeletal changes, in other words, the shape of the cell will change, and they start to move and show great motility. So cancers with aberrant MET activity are highly aggressive, highly invasive, and metastatic, and these patients have a very poor prognosis. So since we saw such nice results with tocotrienols on the HER receptor, which is a receptor tyrosine kinase, we thought it'd be interesting to see what happens with the MET receptor. And so, and just a quick review again, this would be the MET receptor, HGH binds to it, you get dimerization, and then you get the autophosphorylation. So it's very similar and, to the EGF receptor. Now, this is kind of shows you what it looks maybe in a cartoon. You have the MET receptor up here, it binds HGF, and it activates a lot of these signaling pathways, MAT kinase and PI3 kinase, AKT, some of these RAC and RO, which are associated with changing the cytoskeleton and adhesion, motility, invasion, proliferation, cell survival. So the MET receptor is very important in changing the behavior and turning cells very metastatic and invasive. Now, this is basically how it works. There's MET receptors on the epithelial cells, and as you can see, epithelial cells are very organized, they're columnar, they stick to their basement membrane, they don't move, and they have a polarity to them and a regular shape. Now, mesenchymal cells will produce HGF, which will stimulate the epithelial cells, and in this case, increase proliferation, a little more stimulation, and you get a change in morphology, and that morphology will break them away from this basement membrane, and they'll invade tissues, move, and get highly aggressive and metastatic and change their morphology. So that's, HGF has very definite characteristics. Now, one of the first experiments we wanted to do is see if HGF makes these tumor cells that we use grow. And so we use serum-free media, which means there's no serum in it. It's a defined media. And the only mitogen that we put in there is HGF. And in this experiment, you see a very nice dose-response increase in growth. So HGF stimulates these tumor cells to grow. And in the bottom, what you see here is in the absence of HGF, there's no phosphorylation of the MET receptor. Remember, you stimulate the receptor, you get those autophosphorylation. So when you add HGF, within 30 minutes, you get peak autophosphorylation. So everything was working. Now this is showing you what happens with these tumor cells. Again, cytochemistry, this is with an antibody that binds to the phosphorylated MET receptor, the activated MET receptor. So in the absence, all you see is the background staining, and then in the presence of HGF, you see this red on the cell surface. That's the expression of the phosphorylated MET, and it peaks in about 30 minutes. And this is just the negative control. Now, the other thing that's important to understand with tyrosine kinase receptors is that they're very promiscuous. They'll dimerize pretty much with anything that's available. So HER will bind with HER. It also will bind with IGF. It'll bind with the platelet-derived growth factor receptor. It'll actually, you can get heterodimers with HER receptors and MET receptors. So the question was, does HGF actually activate the MET receptor, which will then dimerize with, let's say, a HER receptor, and then the HER receptor will be activated and cause signaling transduction. So it was important to determine if these effects were very specific. And as you can see, in the absence of EGF or HGF, there's very little phosphorylation of either receptor. When you add HGF, there's no phosphorylation of the EGF receptor, but plenty in the MET receptor. And when you add EGF, you get EGF phosphorylation, but not MET. So the effects in the cells were very, very specific, and not due to promiscuity of the receptors, you know. So here's in 3 different cell lines. This is a very metastatic, very invasive mouse tumor cell line. And then MCF7 or human breast cancer cell, estrogen positive. And then the MDA, MB231 tumor line is a highly aggressive, estrogen negative, metastatic human cell line. And as you can see, tocotrienols give a nice dose response inhibition in these cells, and so does this SU11274, which is a specific MET inhibitor, okay? So this inhibits the ATP binding of the MET with the MET receptor and prevents receptor activation and phosphorylation. And so you see these doses, it takes relatively high doses to get this inhibition. Now again, what we wanted to see is look at the combinations at very, very low doses, sub-effective doses. And again, this is more characterization. This is the effects of HGF on MET expression, and then this is the effects of tocotrienol. It blocks the effect. So what we wanted to look at is basically a little dose response here. So we took low doses of tocotrienol. This is in the highly metastatic invasive human cell line. And we used low doses and didn't get an effect. And then we used a low dose of the MET inhibitor, and you can see it, no effect. But when you start combining the MET, sub-effective dose with MET with these sub-effective doses of tocotrienol, you see a very nice inhibition. Now, the graph next to that is called isobulgarian statistical analysis. Is basically you take the IC50 for tocotrienol and the MET inhibitor, and then you just draw this line. And then when you take 2 doses and put them together, if they fall on the line, that means the drugs are additive. If the IC50 of that combination is above the line, it means they're antagonistic. So in other words, they inhibit each other's effect. But when they're below the line, they're synergistic. So as you can see, you take 2 sub-effective doses of tocotrienol and a MET inhibitor, and you get this synergistic anti-proliferative effect. Now, again, we wanted to see how specific this was against cancer. Now, these are 2 cell lines, the CLS1s and the MCF10A. These are basically normal cells. So you have mouse normal mammary epithelial cells and human normal mammary epithelial cells. They're actually immortalized, so they're not totally normal, but they're not tumorigenic. And we use the same doses that we tried in the tumor cells. As you can see, high doses of the MET inhibitor will inhibit the growth. But if you use the same doses when we did that synergistic study, those same doses which inhibited the plus SA mouse cells and the cancerous human cells had no effect. So this was a very specific effect against cancer cells. Against human cells, this synergy wasn't seen. So obviously, if you had much bigger doses, you can get an effect. So the beautiful thing is, at doses that inhibit tumor cells, they had no effect on normal cells. Now, again, this is just more characterization. You see the nice dose effect of the SU11274 and tocotrienol. And again, when you have HGF, no HGF, there's no phosphorylation of MET. When you add HGF, there's phosphorylation. When you add HGF plus tocotrienol, you actually decrease the levels of MET and the autophosphorylation. But when you give HGF and the MET inhibitor, you block the phosphorylation, but the MET levels don't change. So this tells us that both tocotrienols and the MET inhibitor inhibit MET autophosphorylation, but tocotrienols have another action, and that is they lower MET receptor levels. Now, this is, again, using a combination of sub-effective doses. Tocotrienol, the MET inhibitor, you put them together and you get a synergistic inhibition of growth. And again, this shows that when you put, again, HGF, let's see, you get MET phosphorylation, you put HGF with tocotrienol, you block that phosphorylation, HGF with the MET inhibitor, you block that phosphorylation. But when you put them all together, you get a much more robust inhibition. So they are synergistic, and they do have different mechanisms of action. And this is, again, this Ki67. the bottom line is you get nice Ki-67 expression when you use the sub-effective doses, but you put the two sub-effective doses of tocotrienol and the MET inhibitor together, and in the presence of HGF, you really block that autophosphorylation. This is the combined picture that you see the pink dots in it. So it's a very potent anti-proliferative. When you combine these two, it really anti-proliferative. Now, we spent a lot of time looking at signal transduction pathways, and so we wanted to see what was going on. And again, there's lots of pathways, the MAP kinase, STAT pathways, PI3 kinase, AKT pathways. And what we found is, like, if you look at these pathways, basically all these areas here, there's really no difference. When you put them together, you see a nice inhibition of STAT1, inhibition of phosphorylation of STAT5. When you look at on this side, maybe I should show them one at a time. And then if you look on the other side, compared to the control group here, PI3 kinase is down, phosphorylated AKT is down, and phosphorylated or activated NF-kappa B is also down. And this is a phosphatase which can also inactivate NF-kappa B and AKT, in other words, dephosphorylate. I mean, what we're seeing here is this inhibition is due to a decrease in PI3 kinase and downstream signaling, not due to an increase in phosphatase expression or activity. So it knocks out several signaling pathways. Now, getting back to what we were talking about, HGF will cause epithelial cells to change their characteristics, their morphology, their mobility. And they do this by really changing them into, they change them from epithelial cells to mesenchymal type cells. Now, mesenchyme, stromal cells, fibroblasts, that sort of thing, they have a different morphology, they're not very organized, and they're very mobile, and they have some cellular markers that are characteristic. Now, if you look here on epithelial cells, like I said, they're stuck to their basement membrane and very organized in shape. In terms of cellular markers, there's cadherin, and then claudin, occludin, some cytokeratins. But when you have this epithelial to mesenchymal transformation or transition, growth factors will do this, HGF does that. You get epithelial cells to change into something that's more like a stromal cell, a fibroblast. And there's different mesenchymal markers, particularly vimentin, fibronectin. So epithelial cells show high levels of these markers, and stromal cells show high levels of these markers. The difference, too, in the characteristics is, you know, at the bottom here you see this cobblestone arrangement. They're non-mobile, they're non-invasive. Where the mesenchymal transition, you see they're elongated, they're motile, and they're invasive. So one of the things we did was what's known as a wound healing assay. And basically you plate cells on a dish and then take a little plastic pipette tip and just streak it. And you create these wounds. And these things are dying, so you're going to have to bear with me here. But in the first one, you see this wound. It's an open area. And then we treat it with HGF, and the cells grow. And as you can see, within 24 hours, that wound closes, okay? And this is with the sub-effective doses. It doesn't close as much, but it still closes. When we put the sub-effective doses of tocotrienol and the MET inhibitor together, you really block that mobility and wound closure. And so it's preventing the motility of these cells. And if you look at the cellular markers, as you can see here, now this would be in cells grown in HGF and here. And you can see they're, relatively speaking, they have a relatively low level of epithelial cell marker expression and a high level of vimentin, which is a stromal cell cellular marker. But if you look at the, when you give the combination, there's a huge increase in epithelial cell markers and a decrease in the mesenchymal markers. So HGF induces epithelial to mesenchymal transition, and the combination of tocotrienol and the MET inhibitor actually blocks that expression or transition. And this is just a cytochemical study. Again, these are, on this side is in the control, these are epithelial markers, and this is vimentin, which is a mesenchymal marker. And you can see the cells that are grown in HGF show high levels of the mesenchymal marker and low levels of the epithelial marker. But when you go with the combination treatment, the epithelial cell markers go up and the mesenchymal markers go down. So it shows a very nice inhibition of that epithelial to mesenchymal transition. So this is just kind of shows you the MET receptor again. It has lots of interactions with different signaling pathways. You have the MET kinase pathway, the Rho and FAC pathway, which is associated with cellular adhesion, motility, invasion, cytoskeletal changes, the PI3 kinase, AKT-mTOR pathway, which is associated with also proliferation, but also changes in survival, and then the NF-kappa B. So that's the pathway. And basically what this says is tocotrienols inhibit both EGF and MET receptor tyrosine kinase activity, and these results strongly suggest that tocotrienol treatment may provide significant health benefit in the prevention and treatment of breast cancer in women with deregulated HGF-MET receptor signaling. And they're very effective either as monotherapy, but also in combination with other MET inhibitors to inhibit highly invasive and metastatic forms of breast cancer. And these are my collaborators that I've worked with. And this is, like as was said earlier today, we're really in the business of curing cancer in women, not mice. And so we have to make sure that we can, we'll move on to the clinic and see if this can be an effective way for treating metastatic cancer in women. Thank you.
Thanks, Paul, and perfect timing. We've got about 3 minutes, a couple of questions. Anyone have any questions? Please take the mic and identify yourselves. I guess it's all clear. All right, well, if there's no questions, I would like to move on. Thank you, Paul. Our next paper is going to be Tocotrienols in Cellular Aging, Prevention, Evidence from Genes and Protein Analysis. And this paper is going to be presented by Professor Dr. Susanna McPaul.